Good day, colleagues. Thank you for joining us for this webinar on an update on COVID-19 vaccines. This is very timely, as I'm sure you're all aware of the recent announcement on the more than 90% efficacy of the Pfizer-BioNTech vaccine. Our presenter this afternoon will provide some more insights into this important recent development, as well as an update on COVID-19 vaccines development in general. I am Dr. Shanti Singh Anthony, the Knowledge Management Coordinator for the Pan-Caribbean Partnership Against HIV and AIDS, PANCAP, and I welcome you on my own behalf and on behalf of my director, Dr. Rosamond Adams. Thank you all again for being here. This webinar is in collaboration with the Pan American Health Organization. We're pleased that you're able to join us once more as we continue to share knowledge that is currently evidence-based and relevant at this time. COVID-19 has impacted on all of our lives in different ways, and we're monitoring the situation on a daily basis, I'm sure. But just as a really very quick reminder, as of this afternoon, the World Health Organization reported a cumulative total of greater than 50,810,000 infections, with more than 1,200,000 deaths. There's a lot of discussion going on the second wave, especially in countries in Europe and North America, as these countries are entering the fall and winter months. And in the recent weeks, we've seen a spike in daily new infections in those countries. Of course, I think one of the big questions on all of our minds is when will a vaccine become available? And that is what we'll be talking about this afternoon. Before I proceed, let me share a few housekeeping notes. First, we're recording the webinar, and so I take your presence as a no objection to the recording of the webinar. We will share the recording on PANCAP's website and on social media platform for the benefit of persons who are unable to attend this live session. Second, your mics are muted. If you have any questions, as the presentation progresses, please type and submit these in the question field on your webinar panel. We will address them at the end during the question and answer segment. Finally, you will receive a post-webinar evaluation. I encourage you to complete same because it'll provide us with important feedback as we continually strive to improve your webinar experience. At this point, let me introduce to you our presenter. Dr. Jayaweera is a professor of medicine at the Department of Medicine, Division of Infectious Diseases, University of Miami. Dr. Jayaweera brings to this session a wealth of medical experience, having graduated with his medical from the University of Colombo in Sri Lanka in 1976. Since then, he served in numerous positions in Sri Lanka, the UK, and since 1992 is with the University of Miami Miller School of Medicine. Dr. Jayaweera is a member of the Royal College of Physicians, a member of the Royal College of Surgeons, a member of the Royal College of Obstetricians and Gynecologists, and a fellow in the American College of Physicians. He is the current Director of Clinical Trials and Support Services and the Director of Interdisciplinary Stem Cell Institute at the University of Miami. He's an attending physician at the University of Miami Hospital and Jackson Memorial Hospital. Dr. Jayaweera has more than 200 peer-reviewed publications, including 2 recent ones on COVID-19. It is my pleasure to invite Dr. Jayaweera to make his presentation.
Thank you. Thank you, Shanti. Can everybody hear me well?
I think so.
Yeah, thank you. First of all, I want to thank my colleague, Modi Ram Gopal, who is one of your compatriots, who are my very good colleague and friend, who introduced me to Shanti and her husband and the whole of the Guyanese community. And I'm delighted to be here speaking with you on this occasion. I know it's a lot to take and you guys are busy, but I'm grateful that you have joined us. So as I again, I want to thank for the introduction that given nicely. So my task today is to talk to you about COVID-19 vaccines and update. As you can see that the United States have done a terrible job managing COVID-19 due to many reasons, which I don't want to go into politics, but we get a F- for our performance in United States, despite everything. So I came from the HIV world. I'm a HIV researcher and hepatitis C. And when HIV, as you can see that I was there from the beginning of HIV, and when it came, I always thought about Plague by Camus. When Camus wrote the book Plague about the pandemic of plague in North Africa, he exactly described what we are seeing now. They fancied themselves free, and no one will ever be free as long as there are pestilence. And it is so strange that we all knew that this could happen to us one day, but everybody thought that it would happen on somebody else's watch, and it will really not affect us directly. It will happen somewhere in India or Africa or Sri Lanka, but never thought that it will happen in the Western world and Europe. So despite our wealth, technology, the scientific knowledge in the current pandemic, the deaths have been the greatest among the weakest living in the strongest countries. So which is really true. The healthcare system in the United States is so tilted that the vulnerable doesn't have that much support, and the deaths were highest in the group. So, so can you hold on? I'm going to take that phone off. Give me 1 second. So sorry, my office phone was going, that disturbed you. So the major milestones. So when you develop vaccines, so the vaccines first came when Jenner vaccinated his gardener's son. But actually, the vaccine story comes from India. Even about 200 years before that, the Indians were vaccinating because they knew that this was small exposure of bacteria or viruses or the inoculum will lead to development of some immunity which will protect you from the disease. So although they didn't have the immunological background, but they knew that there was something going on. So Jenner, then it came to Turkey. From Turkey, the British picked up, and then the Jenner gets the credit. And then, of course, all the other different vaccines was started. So it started with the smallpox, and then over time, there was a refinement of vaccinations. The live attenuated rabies was in 1885. Then the kill vaccine for cholera and plague and typhoid. And you can see this graph that in 1950, Salk and Sabin developed the polio vaccine, which we all know. Diphtheria came in 1948 or in 1900s. And then over time, we came up with really good vaccines for flu, meningococcal, pneumococcal, hepatitis B, and now first therapeutic vaccine for prostate cancer, and also using reverse vaccinology, you can use meningococcal vaccine. So we have come a long way. Why I'm saying is that because the Pfizer and Moderna has even a different vaccine method, which I will talk later. So when you try to develop a vaccine, what's the strategy? So you start with the platform. You decide what platform you want to use. So say, for example, Pfizer uses the RNA, Moderna uses the RNA, naked RNA, and some uses DNA. I get to that later. But you decide what platform are they using, adenovirus or different methods. And then once you find the target, you prepare a small batch and you do some in vitro testing, whether you can actually neutralize live viruses. That's just the first step in finding out whether it works. And then you look at the target isolation, whether it's the general neutralizing antibodies or what is the antibody that works. And then you validate it with the animal model. Either it starts with mice, and then you can go to non-human primates like monkeys. Once you've got all that done, you go to the phase 1 where you give to humans who are healthy. and see whether there's any response, whether any, just to see whether they are safe. And then you want to find out what the dose is. In phase 2, you want to do the safety and efficacy and the dose. And then you go to the phase 3, where you want to do a large randomized control trials, which I'm doing now with Janssen vaccine, to see whether this really works. So that's not the end of the story. Now, the Pfizer vaccine, they say that it's good. Well, let's see what the data shows when it goes to the FDA. So the two things that we are looking for is the efficacy and safety. Once you know that it works well and it is safe, then you will go on to commercializing it. And then the biggest problem here is the worldwide distribution, scaling up, manufacturing, supply chain. Those are the things that are really not that easy. So this is not that simple. So if you look at the polio took 60 years for us to develop. Ebola took 15 years. But honestly, when we got the Middle East, the MERS, and also the H1N1 pandemic in 2008, we developed a vaccine within like 8 to 9 months. So COVID-19 is not the only a disease that we have done a vaccine development in warp speed. We did it in H1N1 when there was this H1N1 pandemic or epidemic. So we have done it before. So if you look at all the licensed vaccines, there are different methods of production. So this is what I said, you have to create the platform. So either you can use a live attenuated, that means live but kind of weakened, kill whole organisms, or toxins. You take the toxin and you change it and you make it into toxoid. Then a polysaccharide, like conjugate, combinate. There were different methods. You don't have to go into details. But what I'm trying to say here is that the flu vaccine can be in any of these platforms. So similarly, when in next year, when coronavirus vaccines are available, there'll be all different kinds of platforms that are working, and you get to choose which one you want to use and which is the safest or the best, right? So I want to give you a brief introduction between a coronavirus and influenza, because people get all mixed up. So the coronavirus led to SARS, serious acute respiratory syndrome, in 2003 in southern China. Then it caused the Middle Eastern respiratory virus called MERS in 2012. Now we have COVID-19, SARS-CoV-2, which is 2019. Now, the earlier SARS had a very severe, if you get it, 30–40% would die. Not like in COVID. COVID, people don't die, but the SARS didn't spread very much, whereas COVID spreads easily. So it's a kind of two completely different types of viruses that have difference. Whereas the flu, there is the influenza A and influenza B. The worldwide pandemic was H1N1, was influenza A. And influenza A has the H1N1 and H3N2. And April 2009, there was a pandemic of influenza A. Influenza B is there are two genetic lineages. You don't need to go into that. So what happens in flu? I want to complete that before I go to corona. Flu, H1N1 and H3N2, there are two things that happen. One thing is called antigenic drift. So the antigen, the products of the cell or of the virus, not the cell, the virus, that creates antibodies against it when you give it to human or animal. That's the antigen. So the antigens change slowly, called antigenic drift. That's why every year you have to have a separate vaccine. Antigenic shift is when there's a huge change and you almost get a new virus. That's what happened in 2009 when the H1N1 pandemic happened. So there was an antigenic shift. So we pray like hell that this wouldn't happen to any of these viruses. The second thing is how effective is the influenza vaccine. So I want to put these things in context when you talk about the coronavirus. Its full influenza vaccine effectiveness is about 41%. 41%, so it's not that effective. Among people 18 to 64, people over 65, only about 37%. And 18 to 64, only 55%. So flu vaccine is not that great. Flu vaccine doesn't work great when people are over 65. No vaccine works well, when people are over 65, because our immune system is kind of, breaking apart. So, but it is what it is. It's a very democratic system. Whether you're rich or poor, your immune system crashes as you get older. And the important thing is that if this slide here on the left of the age doesn't influence hospitalization rate, who ends up in hospital? This is in America from CDC, is the blacks and the Hispanics and the minorities. So it's very fast stat, because the people who are at the lower end of the scale ends up with this. So here's the coronavirus. I'm not going to go into details. There are multiple proteins, which leads to multiple antigens. So this slide tells you the life cycle of SARS. So the SARS-CoV-2 virus enters. It can enter through anywhere from the eyes, face, mouth, nose, anywhere. Obviously mucous membrane, and then it can go through the angiotensin 2 receptors, which in the lungs, and also it can get into the system through the gut. So it can go through the gut or the other mucous membrane, and gets taken by the cells, and then it gets present to the viral into antigen presenting cell, and the antigen presenting cell will there will be viral if the virus comes in, the viral replication happens, right? And the viral replication happens even any cell with a angiotensin 2 receptor, and then following the antigen presenting cell, the antibodies will be formed, either T cells or B cells. So that's the immune system. So what happened in COVID-19 is that during this activity, there was a cytokine storm. All these chemicals that signal with each other, interleukin 6, interleukin 10, interleukin 1, all these things just shut up depending on the individual, and a lot of people died because of that. So now let's go back to the vaccines. So the vaccines, so there are various vaccines. So the vaccine can be made from the DNA or from the RNA. You can use a non-replicating vector like adenovirus, or a replicating viral vector. You can use a little bit of a protein like spike protein, like the spike protein that the virus used to enter angiotensin 2 receptor, or you can have a live attenuated virus. That means kind of weakened virus. And the doses are like either single dose or multiple dose. So all these viral vaccines is a single dose. So I'm doing the Johnson, is only single dose. The activity is the RNA and DNA activity is very fast. The virus works very fast, and the scale of producing the vaccine production, and manufacturing is generally high in most of the things. In RNA, maybe low to medium, but in general, it's high. So this slide basically says, this is kind of old, but I kept it just to show how companies have worked on this. So now let's talk what are these vaccines available. So the Oxford used a non-replicating viral vector. That means you take the virus, adenovirus, which doesn't replicate. You put a spike protein or one of the proteins from the coronavirus into the virus, and you inject. So the virus goes and infects cells and spreads across the body, and then the cells that get infected will show the antigens on their surface. Now the immune system will come, generate antibodies, cellular B cell antibodies, T cells, and then you get the B cells producing IgG and IgM, and those will finally eventually get rid of the virus when it gets come in. So the Janssen used adenovirus 26, China vaccine uses adenovirus type 5, then the Moderna uses RNA, the Pfizer BioNTech uses RNA just like Moderna. It's a new technique, so which is really good, and then the other like Novavax and different people have uses different vaccines. And the interesting thing is that these things have been tested on other diseases. So except RNA virus, so RNA vaccine. So that's why the Moderna and BioNTech has not been tested before, that technique. But that doesn't mean that it's bad. I don't think it's bad. I think it could be pretty good. So so let's go to the safety and tolerability. So I started off by saying that no matter what you give, you have to have safety, and you have to have tolerability, and you have to have efficacy. I mean, no matter, I make this joke about getting this medicine and vaccine. It's like getting married, you know. I mean, no matter how good the lady is or the gentleman is, if you can't live with it, it's not going to last long, you know. You're not going to take it. So you have to be able to handle this drug. Okay, so here are the side effects. I mean, this is from the Chinese vaccine, and adenovirus vector vaccine. And you know, everybody has pain when you get a shot. I mean, who gets pain when you get an injection? And then, of course, some people, you know, fever. I mean, it's common, you know, with any flu vaccine, everybody gets fever. So these are common things. The only thing is that how bad is swelling and itching. I know one of my friends took a vaccine recently, and that was really bad. Not on the Johnson, but another vaccine. And he said it was horrible. So these things matter, right? So the antibody response were dose dependent, and another thing about the antibody response, that if you had previous adenovirus infection, the response may be a little bit diminished. So that's a disadvantage of the vaccines that use adenovirus. For example, Janssen China vaccine and Oxford vaccine. If somebody has multiple adenovirus infections before, that there may be antibodies against this vaccine, so there may be slight problems. So the Janssen vaccine, they use adenoviral vector, which is not competent. They are not viral replication competent. It has a lot of experience with this, a lot of experience. It has used in HIV, Ebola, RSV, Zika, malaria. They have tested in many diseases. So therefore, a lot of experience. That is the key, because they have perfected it so that you get what you call a Th1 response. The Th1 response is very important, because if you get a Th2 response, I come to that later, you get bad side effects. So they have perfected it. So that's why it is good. So this is from the Pfizer. You can see that so the black column is the one with the convalescent plasma. So what you say is that if you're starting vaccine, you should at least get antibodies as much as convalescent plasma. So and this showed that it got that. Okay, so here are the side effects, pretty much similar. Almost everybody has side effects, but it's, you know, mild. You know, only very few people had severe vaccine side effects in Pfizer. Same thing with the this is from this was the mRNA vaccine from the from Oxford, and these Oxford vaccine with the neutralization response, and pretty much the same thing. This is again another China vaccine, similar response. So you can see now practically. Okay, this is another vaccine, which is again another this is actually the Oxford vaccine, and here is the response of the. So here the green is the convalescent plasma, and here are the antibody response from days from vaccination. So you can see that when you take the Oxford vaccine, you get as much as this. So now which was the Pfizer early data. So the Pfizer said a couple of days ago that they started in July 27th. They enrolled 43,000 people. 38,000 has received the second dose. And they said that the early data shows about 90% effective. They have total 164 cases of confirmed COVID. So now you have to see which one, which is the placebo, which is the real thing. So the thing is that you have to do the final analysis. It has to, this endpoint verification, all that has to be done by the FDA. So you cannot be 100% sure just because a company says it is working well, because they have a conflict of interest. So we will see what the FDA says. Until the FDA says, we are not going to talk about it too much. The vaccine has to be chilled at -80. So both Moderna and Pfizer, they also has to go to -80. I think all the vaccines have the same problem. Okay, so this is what I was talking about. So the safety is the primary goal, and the vaccines can cause what you call antibody-dependent enhancement or vaccine-associated enhanced respiratory syndrome, which means that with Ebola, when they gave it, they found that some people, when they got Ebola, they got worse. I mean, the people who got vaccine when they got Ebola got a worse attack. So that's how they realized that there was a vaccine associated enhanced respiratory disease. So but the advantage of the Janssen vaccine that they have worked with us and they have created a better vaccine. So this is what I was talking about. So there's a very interesting concept called challenge studies. Challenge studies is that now what Jenner did was Jenner took a child, gave the cowpox vaccine, and then exposed the child to smallpox. Now it's a huge risk. You're exposing a child to smallpox, but you found that it worked. So that principle has been worked in many. For example, when they developed an influenza vaccine in 1980, University of Virginia gave the vaccine and exposed them to the disease. And same thing with H1N1, NIH did that, cholera, again the NIH, malaria, the Oxford group did that, typhoid, again the Oxford group did that, and then the dengue, Hopkins did the same thing. So basically give the vaccine and expose them. The why this is interesting is that then the sample size, you don't need thousands of people sample size. You need about 3,000-4,000, and you can get the results very quickly within 3 months. So this can be done because this is what this slide shows. In the United States, there are 30,000 people have signed up for a challenge study, but you know the political climate in US and countries are not very good. But I think this may happen in UK unless the current vaccines work very well. So basically give it, and we use the exposure of the people who are the healthiest. So between 18 and 30, those are the healthiest group, have the lowest mortality with COVID-19, and they can be exposed after vaccination. Just a thought, you know, just a thought. It's something that can be considered. So finally, I come to this, the how you distribute. So once you develop the vaccine, you have to distribute. Now, especially coming from CARICOM countries, you know how difficult it is. So you have to develop the network. You have to create the communication strategy. You have to create a phase 4 evaluation, because we don't know what side effects will come on different ethnic groups. So, and the other thing is that we may be expecting people to take the vaccine, but people may not take the vaccine. For example, when H1N1, they created a lot of vaccine, and lots of people didn't want to take it. So we may end up with millions of vaccine doses. People are not taking it. So the H1N1 vaccine was available in October 2009, and they used the priority groups as the people who are in healthcare and at the lowest this thing. So the lessons learned was that don't get too optimistic about people asking for it. You must get the priority groups. You have to understand their population values. You have to be flexible for the local context, right? And lessons learned from H1N1 should be practiced. So the National Academy of Science in US asked who should get the vaccine. So what they said was, there's a group of experts said that they had to create a social vulnerability index. That's the index that depends on the geography, socioeconomic status, housing density, minorities, but not exclusively race. Similar thing is called the area deprivation index. So those are this, you know, social issues that affect healthcare. So in any country, there is a social vulnerability index. It's not just for US. Every country has a vulnerable population. So you have to take this into account. And then the issue is that you have to prioritize. You have to use the social vulnerability index, but again, you have to prioritize. So this is looking at the pandemic influenza vaccine. They looked at the occupational groups like high-risk population, pregnant women, infant and toddlers. Well, these vaccines have not been tested in these people, so you have to test them and see whether it works. Then deployed personnel, you know, critical healthcare workers, pharmacists, nurses, you know, healthcare workers, janitors in hospitals, and then you go on to the other high-risk population, children, and then you go to other people like over 65, and then finally comes to healthy adults, which is like 132,000 people, 132 million. So if you look at this US population, That's the way you should develop. But we know that rich people always has a backdoor way of getting things. I don't think we can stop that. But at least we can make a pretty good effort so that poor people, vulnerable people, people who need this, who doesn't have a voice and doesn't have money, gets an equal chance of getting the vaccine, and across the world, not just United States, every country, whether it's Haiti or Sri Lanka or India or anywhere, it should be available. So this is the consideration where the CDC came out with. It's pretty much similar what I said. The pandemic influenza framework for vaccine allocation, that was the framework that they were developed. And then the principles of evidence to recommendation and the ethics and equity principles. This is basically going with what we learned from the flu vaccine, adapted for the COVID-19. The whole goal is to minimize death and serious infections, preserve functioning society. So you have to, be able to make the society work. You can't shut down the whole society. You can't shut down schools. You can't shut down factories. You need to, life has to go on, right? And be transparent and be very honest with what we do. And in removing barriers to vaccination, make things easy. Don't make it too complicated. And finally, my last slide in summary. The vaccine will be available next year. I don't think it's going to be available this year, but it'll be next year. They appears to be effective and safe, but details are pending. Who knows what we'll find. The biggest trouble that we had over the last year because of the US politics, this trust in science has gone down. People has great skepticism about science because politicians have muddied the water and used it in very different ways than we are used to. And therefore, the acceptance of the vaccine by the people, we have to do some work. The challenge would be to implementation of vaccination program across the globe and creating a just and equitable solution that at the end, we treat everybody equally as much as possible across the world, not just rich countries. And with that, I want to end this and thank again Dr. Shanti and her husband and all the people from CARICOM and PAHO. And thank you very much for inviting me. And I'll be happy to take any questions.
Thank you so very much, Dr. J. I think that was really very comprehensive. Interesting. I like the way you've touched on some history, taking us back to the days of the plague and really pinpointing and sharing some of the similarities between the plague we're dealing with right now. For sharing the origin of the vaccines, the stages in development, and going back to your last slide that we have to trust the science. I think we have to, because when we think about how many years it took for us to get a polio and Ebola vaccines, now it's only 9 months for a MERS vaccine and H1N1. I think it really is giving us a lot of optimism that this is coming soon. So, but thank you again. I think it was really very interesting. Good update, much food for thought. Like again, the way you've really brought out some of the similarities with previous pandemics and how we're applying some of the lessons learned to the COVID-19 pandemic. We have a few questions lined up, and I'll go right into the questions. I'll go right into the questions. So the first question that I have here, someone is asking, does immunity after getting COVID-19 last longer than protection from COVID-19 vaccine?
That's a great question. Yes, I think so. So that's the whole idea. So when you develop vaccines, you use things called adjuvants. So adjuvants is kind of going to boost the activity of the antigen. And by doing that, you get a better bump on the immune, the antibody titers. And it could be IgG or cellular immunity, like T-cell responses. One of the things that we find is with the COVID infection, unless you get a really severe infection, you don't develop much antibodies. So the convalescent plasma studies, what we found was that out of the 100,000 what is given by Mayo Clinic, only 50% had enough, reasonable antibodies. The others didn't have much antibodies at all. So just because you got infection doesn't mean you got antibodies. If you get a terrible infection, then you will have good antibodies.
Okay. Okay, thank you, Dr. J. The other question, with regards to outcome, and this is with specific reference to the Pfizer vaccine, with regards to outcome, how were people diagnosed as COVID positive? Were they all tested, or was it based on symptoms?
That's a good question. Oh, the studies are defined very, very neatly. So we start off with the symptoms. So what we do is that in Pfizer, all these studies, they give them an app on their cell phone. So, and we do it twice a day. I mean, this is very rigorous for the first week. And then if they develop any, so they have a checklist of symptoms. So if they have any symptoms, they give the list, they check. Then they are given a swab to take. And what we do is that we go and pick up the sample from the patient the same day. So that's for the nasal swab. And then they come back to the hospital next day or the following day. We do a nasal swab, saliva, and a blood sample. And this goes on until the symptoms resolve every 3 days. And all these are tested for coronavirus, do a PCR-based assay. So that's how so if we say it is corona, then it is really corona.
Okay, great. Thank you. Thank you, Dr. J.
Sure.
The next question, how safe is the vaccine for HIV positive patients?
That's also a very good question. It is very safe. I mean, well, all these studies allowed HIV positive people to come on the vaccine. So far, there is no signal saying that HIV positive people are at any higher risk than anybody else. But just a caution that, at least in Janssen, when we enroll patients, their CD4 count has to be more than 350. The viral load has to be undetectable for at least 6 months. And that's it. And then, of course, if there are comorbidities, they're still allowed to come in. So I think it's safe. In general, HIV positive people, their immune response is slightly attenuated, a little less, but overall, the response should be good.
Okay, excellent. So we have a few more questions lined up, Dr. J. But the poll is on. I don't know if you can see the results, but we have 37% of the participants this afternoon that they will take the vaccine. 20% no. 43% are saying maybe. Do you want to react to that?
Yes, can you repeat that? I was looking, I couldn't see the thing, the results. I see the quick.
Okay, so I'll share the results with you in a minute. Let me go back to the questions. So can you comment a bit more on the specific advantages concerning specific advantages in relation to the RNA vaccines?
Okay, that's a very good question. So the advantage of RNA vaccine is very simple. It's very easy to make. Very easy to make, very easy to introduce. And the third thing is that you're not using an adenovirus vector, so it's very safe for handling. Because soon as all these adenovirus vaccines, they are genetically modified by introducing the corona, COVID-19 proteins, RNA or RNA, basically. So as a result, so you have to have a very specific IBs, the biosafety standards have to be followed when you do this study at an institution. mRNA is not a genetically modified thing, so it's just the RNA from using from the virus of similar synthetically made RNA, and so and then the manufacturing may be not that simple, but it's cheaper. So it may be more complicated, but it'll be cheaper. So there are advantages of RNA vaccines, but the disadvantage is that it has to be 2 shots. And we don't know what the downstream effects are. On the same way, the DNA vaccines are similar to RNA, but only thing here is that now you have to use a vehicle to electroporate the DNA into the host genome. So that has other complications.
Yeah. Okay, thank you so much. I see a few participants, you have your hands up. I'd encourage you to type your questions and submit them. I'll now share with you the results of the poll.
Here you go.
Yes.
Yes. So this is, I kind of expected this. So let's put this in context with flu vaccine. So I was looking at the flu vaccine across, I'm just looking at the United States, right? Or even across the world. The people who take the highest, about 50%, are the people over 65. The rest of the population, about 40. Young people, less than 30, right? So I don't know whether this audience is less than 30, but this is maybe 42 is pretty good, because if you're maybe 42%, by the time this is without any results. So your heart is in the right place, right? The 42%. So only 22% said no, right? The question I wonder is why? Is it something that you're worried about? Vaccine will cause something bad? So of course, some vaccines cause bad things. So recently I was asked by a group of medical students, why should young people take the vaccines, flu vaccine? Because these people who are anti-vaccine campaign has said that you get autism and you get Guillain-Barré and you get all these immune mediated diseases because of the vaccine. It is true that occasionally people have had Guillain-Barré. So there was a study done by, I can't remember whether it was CDC or Texas. They did a randomized control study looking at this, not actually a cohort study, looking at whether there's a difference. There was no difference between people who had got vaccine or no vaccine who developed Guillain-Barré. So I think the vaccines are pretty safe, the current. If something is bad to happen, it would have happened even without it. So overall, I think I'm pretty encouraged by this, because that means that almost 78% say they're okay. That's pretty good.
Yeah. Okay. Excellent. So we have a few more questions, Dr. J.
Yeah.
2 and 1. Can a mother pass on passive immunity to her unborn child? If she is vaccinated during pregnancy, does it cross the placenta?
That's a very good question. Yeah, I think some of the antibodies will cross. So there may be some protection, not completely, but some protection. So the baby, but most likely the baby also needs a vaccination. And so your question is, does it protect? So the question is, like, if you get the vaccine and get pregnant, are you safe? We don't allow people to get pregnant for the first month because we don't know, because it's a genetically modified vaccine. But after that, they are allowed to get pregnant. But I think it would be safe. I think it would be safe, and the baby would be protected to some degree, but I don't know how much. But yeah, it's hard to say because we don't have the data.
Okay, thank you. So 2 separate questions from 2 participants, but the same. How safe is the vaccine for asthmatics or people with known allergies?
Very good question. Yes, we don't think that there'll be a cross-reactivity with the allergens that causes asthma versus vaccine, unless you have a specific allergy to vaccine vehicles and things like that. So I think it's safe in asthmatics. We are enrolling asthmatics and people even with COPD in the trial. So we're not excluding asthmatics. We are not concerned about that. What's the second part, Shanti?
People with known allergies. Yeah, same thing. People with known allergies.
Yeah. Unless they have specific allergy to any component of the vaccine, we have no problems.
Okay, all right. Thank you, Dr. J. I see a few more participants have raised their hands. I encourage you to type your questions and submit them in. We have a few questions on the board, a few more questions on the board, Dr. J. Can you comment on the antigenic shift/drift of the coronavirus, and if any, how does it affect the efficacy of the vaccine?
That's a good one. That's a good one. Yeah, that's tough because already we are seeing antigenic drift in corona. It's time will tell. Time will tell. This is the problem. But we think the antigenic shift of corona would be less harmful than H1N1 because the receptor, the domains are more exposed. So I'm not a virologist, but basically my colleagues in virology say that if there was an antigenic shift, still it would be a little less complicated than flu. I mean, compared to like HIV vaccine, it's very difficult to develop HIV vaccine. Obviously, you know that's why people are still struggling. After almost 40 years, we still don't have a vaccine, and corona we have it in a couple of months. So it just showed the structure of the virus, that is so easy to make a vaccine, not so difficult to make a vaccine. So I don't think that. So I think both will happen, but the effect of that on the vaccine may not be that significant. But time will tell. It's so hard. It's a great question. It's these are the things that you have to watch because it could happen very quickly.
Yeah.
Thank you.
A similar question, but phrased differently. What would be the impact of a mutation of the coronavirus on vaccine development?
Yeah, it's the same thing. The mutations lead to antigenic drift and the shift. So those are based on mutations. And even the clinical presentation, we are seeing a change. At the beginning, everybody had respiratory symptoms, and now a lot of people are coming with GI symptoms, and they're not that many people going on ventilators. I mean, we are not ventilating that many either, but the respiratory symptoms are much less now. So there is a clear shift. I don't know whether it's the virus or something else, but there is a difference.
Okay. So I'm seeing quite a few more raised hands from our participants. Again, please submit your questions. If you're having difficulty finding where to submit your questions, you can send them in the chat to me. But we have a few more questions on the board, Dr. Jane.
Sure.
Is the vaccine reserved for non-exposed only, or can it be used post-infection irrespective of developed immunity?
That's a super question. That is super question. Yes, of course. Yes. In fact, we are enrolling people who already have had coronavirus infection. So that comes to the initial question. If you had coronavirus, do you need a vaccine? Yes, that just underscores my point that even if you had coronavirus infection, I would take the vaccine because you will have a better immune response, because we know that some people have got reinfected because if you track the antibody titers over time, they slowly go down. So that is that's why that we should have the vaccine.
Okay. Based on current understanding, for how long do you average that a vaccine can offer protection?
That is good. You guys are asking really good questions, So so we are not sure. So we are going to monitor these people for 2 years. So we will have a decent answer in 2 years, at least in Janssen. I'm sure all the vaccine studies are going for 2 years. So we will track these antibody titers. So we see them every 6 months. So initially every 3 months. The second year is, I think, every 6 months, So we will know. So it's hard to say. Because when you do vaccines, if you give a high dose or 2 doses, you may have a longer duration vaccine. But if you give 2 doses or high doses, the side effects are more. So you have to balance between the devil and the deep blue sea. You have to balance between safety versus efficacy and durability. So if we find that vaccine efficacy goes down in about 1.5 years, maybe the solution would be to give a booster shot, like, tetanus, give a booster shot. So that's good. Thanks.
Yeah. Thank you, Dr. J. The next question, what is the expected reaction to a person who has had COVID virus and still has side effects from the infection if they're given the vaccine?
So we haven't seen much. No, we haven't seen. Because I think when you get the infection, you have these cytokines, going crazy. And with a vaccine, you don't have that cytokine storm that people get. So it's a very controlled infection you're not getting the whole virus, you're getting a spike protein antigen through an adenovirus. I mean, they don't tell us what protein they're using because those are proprietary secrets by each company. But whatever they are putting in the adenovirus, it doesn't cause the cytokine storm what a COVID-19 infection would cause. So one doesn't expect this whole, show of symptoms. So we don't expect anything.
Okay, great. One final question before we close. What is the risk of someone having secondary illness?
Secondary illness due to vaccine. So yeah, so those things are hard to predict. So I mean, like, for example, Oxford vaccine, they said that 2 people had neurological problems. One had multiple sclerosis, the other one has transient myelitis. So multiple sclerosis had nothing to do with the vaccine. They established that. Transient myelitis, they say that they're still exploring. So, if you give even normal saline to 300,000 people, I'm sure somebody will have some problem. It is what it is. So you can't live like that. I mean, so I don't think that there will be such a. I mean, it can happen, but the odds are very low. That's what I think. But we will have more information when all the studies are done.
Okay. I wanted to close, but I'll take these last 2 questions because I think they're so and they're both linked to the minus 80 degrees storing of the vaccine. So the question really is whether the person heard correctly and how do you store at minus 18? Minus 80?
Yeah. Minus 80. So they'll have to have special freezers. So in all our labs, we have minus 80 freezer. That's, most of the labs, I'm sure in Guyana, in Trinidad, Jamaica, they have minus 80 freezers. But the problem is, I mean, those are in like specialized labs for preserving, tissue and things like that. But to get it out and in the community is not easy. And then once you thaw, you have only 4 hours to give the vaccine. So once you take it out, in the fridge, at least Janssen vaccine, we can keep it for like 20 or 24 hours. So that is not much leverage we have here to manipulate things. So now the US government is insisting all these hospitals buy a minus 80 freezers to store the vaccine so that they can serve the community. So that's going to be a problem. So here comes the other thing. So there are other vaccines being developed, like the polio vaccine, which are not injectable and which could be given orally. So this story is not over yet. So these are the list of vaccines that are coming out. The second generation vaccines will be developed, which doesn't need that minus 80. That's my prediction, that minus 80 centigrade. That's really cold.
Yes. That is very cool, and I like your prediction that the second wave or the second generation of vaccine may not need -80.
Yeah.
So I would want to again thank you, Dr. J, for an excellent presentation and for excellent discussion responses to the questions. I think we've had a fantastic audience with some excellent questions. Good thinking on the science and on vaccines and vaccine development. So on behalf of all of the participants, Dr. J, I want to say thanks again very much for sharing your knowledge, your experience, your practical hands-on experience that you have with these clinical trials and with vaccine trials. I think we had a 1 hour that was really well spent. To all of the participants, thank you again for staying with us. We trust that you would have gained new knowledge, that you would have a better understanding of COVID-19 vaccine development process, and what's to come. As Dr. J said, there is much hope and there is good enthusiasm. Let's see, next year will bring good things for all of us. So, just a few points before I close. I will be sending the presentation to all of you via email. And a quick survey reminder, you will receive a very short survey seeking your feedback on this webinar. It is important that you provide that feedback to us. It will for sure allow us to better understand and address your knowledge needs, everyone in this region, especially at this time when we're in the middle of a pandemic. So I encourage you again to take a few minutes and provide us with your feedback. And in closing, I also encourage you to stay engaged with us. Visit our website, www.pancap.org, for more information on HIV and COVID-19. In the region, we have a dashboard on our homepage where you can get daily updates on new infection in the region. And we have an entire COVID-19 webpage. Please also follow PAHO. There's a host of resources on the Pan American Health Organization COVID-19 page, from policy to guidelines to protocols, a host of information that will be useful to you. We have a follow-up to this webinar where we'll talk about how can we access the vaccine as countries? How can we access the vaccines? And this is really talking about the COVAX mechanism. So thank you again. I hope you join us for the follow-up webinar. Thank you again, colleagues, for being with us this afternoon. Please stay tuned. Good afternoon, everyone.
Good afternoon.
Thank you, Dr. J.